Hi, students. After a long day due to the lockdown in our country due to COVID-19 pandemic, we meet virtually on social media. Now we know that the situation in the world due to the pandemic COVID-19 caused by one of the positive agent coronavirus. So study of that topic is most important to us as a biotechnologist. We study that topic in four sections. In section first, we learn about the coronaviruses. In second, about the COVID-19. In third, epidemiology. And in fourth, drug discovery strategies for COVID-19. Today we are going to learn about the section first, coronaviruses. First of all, we look on the classification of coronaviruses. Viruses classification is the process of naming viruses and placing them into a taxonomic system. It is similar to the classification of cellular organisms. Robivirus, it is a kingdom of viruses that encompasses all RNA viruses and virions that replicate by means of RNA-dependent RNA polymerases. Incerticides, or it is also called as problematica. These are terms used for a taxonomic group where its broader relationships are unknown or undefined. Nidovirales, it is an order of viruses with animal and human hosts. Coronaviridae is a family of enveloped positive sense single-stranded RNA viruses. And subfamily Orthocorovirinae, they are enveloped viruses with a positive sense single-stranded RNA genome and a nucleocapsid of helical symmetry. Now next, we study the discovery of coronaviruses. Coronaviruses were first discovered in the 1930s when an acute respiratory infection of domesticated chickens was shown to be caused by infectious bronchitis virus IBV. In the 1940, two more animal coronaviruses, mouse hepatitis virus MHV and transmissible gastroenteritis virus TGEV were isolated. Next, human coronaviruses were discovered in 1960. The earliest ones studied were from human patients with the common cold, which were later named. Other human coronaviruses have since been identified, including SARS-CoV in 2003, MERS-CoV in 2012, and SARS-CoV-2 in 2019. Most of these have involved serious respiratory tract infections. Nowadays, COVID-19 is also from due to that type of viruses. Now we see the morphology of coronaviruses. Coronavirus virions are spherical with diameter of approximately 125 nanometer. The most prominent feature of coronaviruses is the club-shaped spikes, projections emanating means outwards from the surface of virion capsids. These spikes are a defining feature of the virion and give them the appearance of a solar corona, prompting the name coronaviruses. Coronavirus is a virus particle contain four main structural proteins. These are spikes S, membrane for M, envelope E, and nucleocapsid N proteins, all of which are encoded within the 3' end of the viral genome. Now we learn about the life cycle of coronaviruses. The initial attachment of the virion to the host cell is initiated by interaction between the S protein and its receptor. The S protein receptor interaction is the primary determinant for a coronavirus to infect a host species and also governs the tissue tropism of the virus. Tissue tropism is the cells and tissues of the host that support growth of the particular viruses. The next step in the coronavirus life cycle is the translation of the replicase gene from the virion genomic RNA. Viral RNA synthesis follows the translation and assembly of the viral replicase complexes. Viral RNA synthesis produces both genomic and subgenomic RNAs. Subgenomic RNAs serve as mRNAs for the structural and accessory genes which reside downstream of the replicase polyproteins. Following replications and subgenomic RNA synthesis, the viral structural proteins S, E, and M are translated and inserted into the endoplasmic reticulum. These proteins move along the secretory pathway into the endoplasmic reticulum, Golgi intermediate compartment. There, viral genomes encapsulated by N protein bud into membrane of the ERGIC containing viral structural proteins, forming mature virions. After assembly, virions are transported to the cell surface in vesicles and released by the exocytes. After that, exocytosis, well developed or mature virions are outward from the cell and infect another cells. Thank you.